Good morning, students. Today we are going to study about the next disease in the chapter Human Health and Diseases, and this is the malaria. So malaria is caused by the parasite Plasmodium, that is protozoan, and it belongs to the family of sporozoans. So it is the protozoa that multiplies asexually as well as sexually in its main host, and it is having diagenetic life cycle. That means it has two hosts. The host is Anopheles mosquito, female Anopheles mosquito, as well as the humans. So its life cycle need to have two hosts for its completion. So now we are going to watch a video which will give you a brief idea about the life cycle of mosquito, and then we are going to about the life cycle of Plasmodium, and then we are going to read within the NCRT that what is given in the CBSE, and what we have to learn about the parasite as well as the disease malaria. So here is the
When a female Anopheles
video.
mosquito penetrates human skin to obtain a blood meal, it injects saliva mixed with an anticoagulant. If the mosquito is infected with Plasmodium, it will also inject elongated sporozoites, motile spindle-shaped asexual cells, into the bloodstream of its victim. The sporozoites travel to the liver, where they enter liver cells and rapidly divide asexually. This asexual division, which is called schizogony, generates the next life cycle form, called merozoites. The released merozoites invade other liver cells and enter the host's bloodstream, where they invade erythrocytes. Once inside the erythrocyte, the merozoite begins to enlarge as a uninucleate cell termed a ring trophozoite. The trophozoite's nucleus then divides asexually to produce a schizont which contains several nuclei. The schizont then divides and produces mononucleated merozoites. The erythrocyte ruptures and releases toxins throughout the body of the host, bringing about the well-known cycle of fever and chills that is characteristic of malaria. Plasmodium enters a sexual phase when some merozoites in the erythrocytes develop into gametocytes, cells capable of producing both male and female gametes. Erythrocytes containing gametocytes do not rupture. Gametocytes are incapable of producing gametes within their human hosts, and do so only when they are extracted from an infected human host by a mosquito. Within the gut of the mosquito, the gametocytes form male and female gametes. The resultant diploid zygotes develop within the mosquito's intestinal walls and ultimately differentiate into oocysts. Within the oocysts, repeated mitotic divisions take place, producing large numbers of sporozoites. These sporozoites migrate to the salivary glands of the mosquito, and from there are injected by the mosquito into the bloodstream of a human, thus starting the life cycle of the parasite again.
So this is about the life cycle of mosquito. What you will now you will be looking at the another screen which will show you the NCRT image. So the image is right now, it's not available. So we are going to discuss the next um, disease, that is the amoebiasis. So amoebiasis or amoebic dysentery is caused by a um, parasite which is known as Entamoeba histolytica. So it is also a protozoan disease. So now we are going to understand what about the Entamoeba histolytica and how the disease spread, what is the transmission, what are the symptoms. So please have a look on this video and you will know about the Entamoeba life cycle.
Entamoeba histolytica is a protozoan parasite that, in most cases, harmlessly resides in the colon. However, it has the capability to cause dysentery, colitis, and liver abscess. The World Health Organization, the WHO, considers amoebiasis to be one of the major health problems of developing countries, surpassed only by malaria and schistosomiasis, or death caused by parasitic infection. Approximately 100 million annual cases of amoebic dysentery, colitis, and liver abscess result in 100,000 deaths occurring worldwide. These infections are found mainly in developing countries such as India, Africa, and parts of Central and South America, where sanitation methods are not as highly developed. However, infections are also an issue in developed countries, with patients usually being migrants from and travelers to infected areas. The first proven case of amoebic dysentery was made in 1875 by Fyodor Loesch in St. Petersburg, Russia, where he originally called it Amoeba coli. This organism was later renamed Entamoeba histolytica by Fritz Ch-7, a German zoologist, in 1903. Ch-7 was the first to differentiate between Entamoeba histolytica and its harmless counterpart, Entamoeba coli. He later died in 1906 when he underwent emergency surgery due to an amoebic abscess from a self-inflicted infection. The first case of North American amoebiasis was reported by Sir William Osler in 1890 after he observed amoeba in stool and abscess fluid from a physician who had previously resided in Panama. The life cycle of Entamoeba histolytica begins when cysts and trophozoites, which are immature cysts, are passed through the colon and expelled in feces. The cysts are typically discovered in formed stool, with trophozoites being found in diarrheal stool. Infection then occurs when an individual ingests mature cysts through fecally contaminated food, water, or hands. Once the cyst is ingested and reaches the small intestine, the contained trophozoites are released and continue to travel to the large intestine. Once they have arrived at the large intestine, the trophozoites multiply through binary fission and produce cysts. Both the cysts and the trophozoites are then passed through the intestine and expelled in the feces.
Now we are going to understand about the next disease, ascariasis, and their symptoms and the organism that causes the disease. This is also as a practical in your class 12th biology as disease causing organisms, their slides, their specimens, and to write their identifying features and also to tell the symptoms of the disease which they cause. So you have to learn particularly in a very important way, because it is important from theory point of view as well as from the practical point of view. So ascaris is the roundworm, and it causes the intestinal infestation, and it causes the intestinal diseases, and the growth of ascaris in large amount in the human can also cause the perforation of intestines. So it is a parasite, it is a roundworm, The features of the ascaris worm you will be able to look in this video, and also about the disease you will be understanding.
Roughly 1.5 billion individuals are infected with this worm, primarily in Africa and Asia. Ascariasis is an epidemic in the United States, including Gulf Coast, in Nigeria, and in Southeast Asia. One study indicates that the prevalence of Ascariasis in the United States is about 4 million, which is 2%. As of 2010, it caused up to 2,700 deaths, down from 3,400 deaths in 1990. Oscarous Lubroids can live 1 to 2 years, and they are the largest intestinal nematode affecting humans. Females averaging 30 centimeters in length. Males are the smallest, ranging from 15 to 30 centimeters in length. Both sexes have an elongated centrical body, which tapes at the top of the ends. In males, the tail curls ventrically. Females have vulval openings. Both sexes are cream-colored, sometimes with a pink tinge. Oscarous Lubroids lack circular muscles, the only muscle bands being longitudinal, and the worm uses muscular activity to remain in the intestinal lumen of the host. This roundworm also lacks a circulatory system, and its digestive, excretory, nervous, and reproductive systems are all suspended with this pseudosolum. Other physical features are they're ectodermic, heterothermic, bilateral in symmetry. Adult worms live in the lumen of a small intestine. A female may produce approximately 200,000 eggs per day, which are passed with feces. Unfertilized eggs may be ingested, but are not ineffective. Fertilized eggs embryoconate and become ineffective after 18 days to several weeks, depending on the environmental conditions. After the infective eggs are swallowed, the larvae hatch, invade the intestinal mucosa, and are carried to the portal, then the systematic circulation to the lungs. The larvae mature further in the eggs, about 10 to 14 days. Penetrate the alveolar walls, ascend in bronchial tree and the throat, and are swallowed. Upon reaching the small intestine, they develop into adult worms. Between 2 and 3 months are required from ingestion of the ineffective eggs of oviposition by adult females. Adult worms can live 1 to 2 years. The Oscarous Lubroids can cause ascariasis. Modes of transmission. Ascaris live in the intestine, and the ascaris eggs are passed through feces of infected persons. If the infected person defecates outside, near bushes, in a garden, or field, or if the feces of the infected person is used to fertilize as fertilizer, eggs are deposited on soil. They can then mature into form is infected. Ascaris is caused by ingesting eggs. This can happen when hands or fingers that have been contaminated on them are put in the mouth, or by consuming vegetables that have not been carefully cooked, washed, or peeled. Infected with ascariasis have no symptoms.
So this is about ascariasis disease, and you can study all these diseases in NCERT as well, because I'm not having the pages. You can study by the video. I hope you have understood these diseases and you study. these through the NCERT, and if there are some doubts, then you will ask me. There is further a disease known as filariasis, which is also called by a roundworm, caused by a roundworm called Malaria, and it is causing the inflammation of lymphatic system. It grows inside the lymphatic system, and mainly it grows in the lymph vessels of the lower limbs. So the limb becomes so much swollen, and with the inflammation, with the action of the WBCs as well as the other immune system reactions against those worms, the leg or the limbs become so much swollen that that condition is known as elephantiasis or the elephant foot. So the image is also given in NCERT. That is the roundworm disease. Then later on, it is given the fungal diseases. So few fungus names are given in the paragraph, like the epidermophyton, microsporum, and these their slides also there in the lab. You can see the slides, and with this you will know the structure of the mycelium of the fungus. So mycelium is the network of the filaments which is making the fungal body, and their spores. The spores and the mycelium features are the important identifying features of their that parasite or that fungal invader. And the symptoms are that you can have a dry skin, lesions can be there in the skin. It can affect some of the fungus can affect the scalp also, and it causes itching, burning sensation, redness, rashes, and certain transmission is generally by infected person through the use of that infected person materials. If we comb by the same comb hair by her the infected comb, and if we use any towel or anything material, personal belongings of that person, so we can also catch the skin disease of that person. So this is the way some of the fungal diseases are also called ringworms, like because the lesions are round in appearance on the body, the fungus make the round lesions. तो जहाँ पर भी फंगस का इन्फेक्शन होता है वहां पर एक लाल चत्तेदार डॉट्स जैसे रैशेस हो जाते हैं, तो इस वजह से उस रैश के शेप की वजह से उसको रिंग वर्म्स कहते हैं, या रिंग कटर करके आपका एक एड भी आता है जिसमें वो दवाई देते हैं जिससे वो फंगल प्रॉब्लम, दाग, खाज खुजली वाली वो प्रॉब्लम खत्म हो सके। तो ये फंगल बीमारियां हैं। तो जहाँ पे भी थोड़ी नमी होगी बॉडी में, और अगर अपनी पर्सनल हाइजीन पे कोई ध्यान नहीं देता या किसी इन्फेक्टेड पर्सन का सामान यूज कर लेता है, तो उसकी स्किन प्रॉब्लम भी दूसरे हेल्दी इंडिविजुअल को हो जाती है। तो ये आपके एनसीईआरटी में दिया है। देन ये आपके सारे डिजीजेस थे। स्टार्ट हमने किया था टाइफॉइड से, जो कि एक बैक्टीरियल डिजीज था जो इंटेस्टाइनल बैक्टीरियाज की वजह से होता है, और विडाल टेस्ट से उसका डायग्नोस्टिक होता है। देन हमने बताया था अबाउट निमोनिया, दैट इज ऑल्सो बैक्टीरियल डिजीज जिसमें कि बैक्टीरिया एल्वेलाइज में ग्रो करने लगता है, और वहाँ पर इम्यून सिस्टम के रिएक्शन की वजह से पस फिल हो जाता है एल्वेलाइज में, और पानी भरने की वजह से, वहाँ पर पस भरने की वजह से, डेड सेल्स होने की वजह से the air exchange affinity ability alveoli की reduce हो जाती है, जिससे breathlessness in the अंदर ऑक्सीजन की कमी हो जाती है, ब्लू लिप्स हो जाते हैं, स्किन ब्लू होने लगती है। तो ये निमोनिया के सिम्टम्स हैं। देन हमने पढ़ा बैक्टीरिया की बहुत सारी अदर डिजीज भी हो सकती हैं, एंटीबायोटिक से बैक्टीरिया की डिजीज किल हो सकती हैं। देन वायरल प्रॉब्लम हमने राइनो वायरस से होने वाला कॉमन कोल्ड पढ़ा नोज में जो होता है। फिर आज हमने जो पढ़ा है मलेरिया की लाइफ साइकिल देखी, वीडियो देखी, थ्रू वीडियो हमने एंटामीबा हिस्टोलाइटा के बारे में पढ़ा एंड एस्करियासिस के बारे में पढ़ा। तो आई होप ये टॉपिक सबको क्लियर होंगे एंड यू ऑल्सो गो थ्रू एनसीईआरटी। थैंक यू। आई वांट टू एंड दिस सेशन नाउ। Keep watching and studying online, and please, happy reading. Thank you.